talked last time about T-cell trafficking or other immune cell trafficking as being one of the events that makes the adaptive immune response take a while. And we're going to see sort of the next step of that process today and continuing on Wednesday, where we actually look at the way that our T-cell is going to become activated. For our T-cell to become activated, it needs to be in the secondary lymphoid organ, and it needs to be in the presence of an antigen-presenting cell and some antigen. And so we mostly were talking about how all of these components get to the lymph node. Previously, I'm going to spend a little bit more time talking about the antigen-presenting cell today, but most of what we'll be saying is about how that T-cell and antigen-presenting cell actually interact once they are together in that secondary lymphoid organ. So as I said, we've got to get the antigen and the antigen-presenting cell into the lymph node. Our antigen, our free antigen, these red dots, can move through the lymphatic system as free antigen quite easily, but very frequently our antigen can also be taken by our antigen-presenting cell. Most of the time, this will be a dendritic cell. I told you before that dendritic cells were the #1 best cells at turning on T cells. We'll see more of that today and a lot more of that. That's going to be a big deal on Wednesday. There are dendritic cells that live in many tissue types that can acquire antigen and then move to the lymph nodes themselves. In our tissues, we have immature dendritic cells. These are cells that are closer to being the early monocyte precursor. So we will have lots of dendritic cells, precursors in tissues, or immature dendritic cells, which are shown on the upper left here. You can also see sort of immature dendritic cells on the top of this slide, or on the left in this figure from your textbook, we have both an immature dendritic cell and an immature macrophage. When that cell comes in contact with a PAMP, when that cell gets a PRR stimulus, this is especially true of our dendritic cell. This also can be the case of getting some cytokine stimulation as well. So when we have PAMP and cytokine stimulation, our dendritic cell is going to mature. It's going to become a mature dendritic cell. That cell is going to undergo morphologic changes. So you can see in the microscopy images, this cell looks really different. One thing that you will notice is that the cell has all of these protrusions on its surface. You would never find a cell like that in the blood because those protrusions would get sheared off by blood flow. The only cells you find in the blood are really nice and round. That would get destroyed. But this dendritic cell can be found in tissue once it's been activated. And that's both PAMPs and cytokines, as your textbook says. When that activation happens, when our dendritic cell in the tissue comes in contact with some type of microbe, a number of different things happen. So our early dendritic cell, our immature dendritic cell, is really, really good at one specific job. That immature dendritic cell has one thing it's really, 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 really good at. And that one thing is phagocytosis. So our tissue dendritic cell is really, really good, lots of plus signs, at phagocytosis or other types of internalization. So our early dendritic cells are basically just taking in things around them. When that dendritic cell becomes activated through PRRs, it's going to stop being phagocytic, but it's going to start making huge numbers of additional proteins. It's going to make way more MHC. it's going to make way more other proteins that can be involved with T cell stimulation. So we're going to see very high MHC expression. We're going to see a lot of delivery of other types of T cell stimulation molecules, our co-stimulators. So once we have our mature, non-phagocytic dendritic cell, there's going to be lots of co-stimulation delivery. And these cells are going to express trafficking molecules. So now instead of hanging out in the tissues, they are going to move through the lymphatic system to a lymph node. So basically, once the cell has come in, basically just going to be trying to pick things up at the beginning. Once it picks up a microbe, it is immediately going to become really good at activating T cells and also move to the lymph node where it can find a T cell. And you can see that process here. So at the beginning, our dendritic cell is going to be in some type of tissue. It's an immature dendritic cell. If it phagocytoses something, it will start making lots of new proteins. It will also start moving to a lymph node. That's partially due to things like CCR7 expression. And that dendritic cell will now be able to potentially activate T cells in that lymph node. So we've got our antigen and our dendritic cell there. The dendritic cell is probably carrying that antigen that it has phagocytosed to present to a T cell. Yeah, Marina.
So I know you said it just moves to the lymph node. That's not through the vessels, right?
It's through the lymph vessels, not through the blood vessels. It's through the lymphatic system. Our T cells get to the lymph node through the processes that we talked about last time. And once our T cell makes its way into the lymph node, once it leaves the HEV, the T cells will actually follow a chemokine gradient to go to the T cell area, which happily is where the dendritic cells are hanging out. So finally, we've got our T cells and our dendritic cells in the same location of the lymph node. And they finally can potentially meet up so that the dendritic cell can interact with the T cell and do some activation. When our T cells in the periphery are activated, they will interact with an antigen-presenting cell like a dendritic cell to see antigen, and that will lead to a number of different changes in that T cell. One of them is that T cell is going to get activated through some signaling. We're going to see cell division, known as clonal expansion. So instead of having one cell, we'll have many. So we'll see expansion of that cell, and we'll see differentiation of that cell. So that cell is going to gain the ability to do some new functions. All of these things are then going to allow that cell to go out and get rid of pathogens. We are mostly going to focus on this step today. How the cell is actually seeing antigen and how that works at a molecular level. And then we're going to move through the rest of this on Wednesday and start into some of these effector functions on Wednesday. So we're really kind of starting at the beginning here, at that signaling process. Two kind of little pieces of information I want to tell you. One of them is that the real story of activating a T cell is that T cell activation requires multiple signals. Every immunologist talks about signal 1 and signal 2. Sometimes we talk about signal 3. We don't always call it signal 3. Signal 1 and signal 2 are always called signal 1 and signal 2. But we do always need that additional thing as well. So we need signaling through a T cell receptor in order to activate our T cell. We're going to need signal 2 through co-stimulatory molecules to activate our T cell. And we're also going to need some interactions with cytokines to turn on our T cell. Today is only going to be signal 1. For pretty much the rest of the day today, we're talking about signal 1. But to give you full disclosure, these other signals are incredibly important. And we're going to see signal 2, which only, won the Nobel Prize this year, on Wednesday. Part of the reason why I mentioned this is actually related to what we saw on this previous slide. One of the cytokines that is particularly important is a cytokine known as IL-2. And you can see IL-2 showing up pretty early in this scheme. IL-2 is going to be important sort of during activation, and it's going to be really important for this clonal expansion part. It's one of these first early steps. And the reason why I'm pointing all of this out is that our goal today when talking about signal 1 is we are going to talk about how we get from signal 1 to the T cell producing IL-2, because the T cell actually produces IL-2 and feeds itself. And so we're going to sort of consider how signal 1 works in terms of how you get from here to that T cell producing IL-2. So our goal at the end today is to make IL-2, which will be important for the next set of steps for that T cell in its life. And really that IL-2 is one of many cytokines that can be important for this thing that's shown here as signal 3. Big picture wise, again, if you want to transcribe the IL-2 gene in order to make the IL-2 protein, and IL-2 is an important cytokine that we'll see a lot more on Wednesday, you actually need to have 3 different transcription factors working together. So IL-2 transcription requires the activity of 3 transcription factors. One of them is called NFAT, NFAT. One of them is called AP1, and one of them is NF-kappa B. So if you want to turn on IL-2, you need to get NFAT, AP1, and NF-kappa B working together. And so again, our goal as we look at signal transduction today is to think about how the signaling we talk about gets us NF-kappa B, NFAT, and AP1. So that's kind of, again, this is the outcome that you should be looking for in this process. Signal 1 is largely coming through the T cell receptor. And so you can remember the T cell receptor that we saw here, along with its partner proteins, CD3, and the co-receptor protein CD4 that's shown here, or CD8 if this was MHC class 1 and a CD8 positive T cell. I also told you that this is not a particularly strong interaction. Our T cell is not able to bind very strongly to our dendritic cell. And in fact, if it was just about this interaction, the T cell and the dendritic cell would not be held together long enough for signal transduction to occur. Fortunately, there are other proteins that help hold the cells together. And so outside of the T cell receptor MHC interaction, there are a number of other proteins that are actually holding that T cell very closely next to an antigen presenting cell. These are adhesion molecules, and largely they are integrins. They're the same integrins that you saw as the adhesion molecules stopping the T cell last time. So the same integrins are also really important for holding our T cell next to our dendritic cell for a longer period of time. In reality, there are many proteins that are on the surface of our T cell and our antigen presenting cell and that are interacting. You can see this from your textbook here. One thing that's sort of interesting is the way that these proteins actually organize themselves in the membrane of the cell. So people have spent a lot of time looking at how these proteins are organized in the membrane of the cell. And what they've seen is that a lot of the proteins that are important for kind of transducing that primary signal end up getting kind of bunched together. And we can actually do some imaging and find out that all of those proteins that are involved in T cell receptor signaling, like the T cell receptor, like CD3, like the co-receptor CD4 or CD8, all come together in one spot on the T cell receptor, on the T cell, in order to form almost like a little target or a bullseye to interact with that dendritic cell. And the adhesion molecules all form an outer ring around them. And so again, if we actually look using fluorescent signaling or fluorescent imaging at where these proteins are, they form sort of concentric circles around themselves. And so here you can see the same thing. At the top, you're sort of looking at a cut through of the APCs at the top and the T cells at the bottom. Here you're the APC looking down at the T cell. And you can see that some things like our integrins are around the outside, whereas our T cell signaling proteins make a nice little bullseye in the middle of the target. So this is sometimes referred to as the PSMAC and the CSMAC. The CSMAC is the central supramolecular adhesion complex, and the PSMAC is the peripheral supramolecular adhesion complex. This was also shown in the previous figure as well. Those were both labeled. And so these proteins are actually doing their signaling process in a very regulated fashion. When this whole thing was described, it was described as something called the immunological synapse. And sometimes there's some competition between immunologists and neuroscientists, like who has the most complicated system. And I think immunologists were, feeling a little feisty because they decided they wanted a synapse too. Immunologists love the word synapse. And realistically, it is being used correctly. A synapse is just any place where two cells come together and interact. It's a cell-cell junction. So this is the immunological synapse, but as we talk about T cells, we like to talk about the infectious synapse and the cytotoxic synapse and this synapse and that synapse. But this is really the first time immunologists started having a synapse, was when we actually described the specific organization of all the T cell receptor proteins when interacting with an antigen-presenting cell. One of the ways this organization works, which I think is really cool, is shown here. And so what you can see is that our cells are basically little circles. And so if you put two circles next to each other, in the area where they're close together, there's relatively small amount of space between them. And as we get further and further from the contact site, there's more and more space. And in fact, all of these proteins differ in their their extracellular domain. And so in fact, the ones that are kind of short are all the ones that are involved in signaling, and so they can fit in this junction where the two cells are close together. The ones that are involved in adhesion are a little bit longer, and so they can make a nice circle around in areas where the cells are not quite as close together. But in fact, there are even some other proteins that turn off signaling. The most famous one is called CD45RA. CD45RA has a gigantic extracellular domain that cannot fit in that junction at all, and so it gets shoved out of the way when the cells come close together. So suddenly we can't turn off signaling anymore because there's no room for CD45RA to be here. This cytoplasmic domain is going to be the inhibitory one, but it can't be near here. There's not nearly enough space when these cells come closely together. So the organization of these proteins is pretty important and it's pretty cool. There's been a lot of really great imaging work that goes on with this. So, here's the beginning of the signaling process. I refer to it at the top of the slide as receptor proximal signaling. Receptor proximal signaling means near the receptor. It's the top of the process. And you will sort of see why this is important later. And the receptor proximal signaling involves some proteins that you've already learned about. So I've already told you about the T cell receptor. We've talked about how the T cell receptor is kind of a problematic receptor in that it doesn't really have a cytoplasmic domain. And so the T cell receptor associates with the additional proteins from CD3, which have these nice long cytoplasmic domains. And those cytoplasmic domains include these little yellow boxes, which are the ITAMs, the areas where we can phosphorylate CD3. So that's the ITAM, the immunotyrosine-based activation motif. And so we have places where phosphates can be added, just because it will be interesting later. The specific amino acid in an ITAM that gets phosphorylated is YXXL. You'll see why I mentioned that later. CD4 or CD8 associates very closely with a kinase. When it's CD4, then it's LCK. When it's a CD8, it's a similar related kinase in the same family. You can say it's LCK for our purposes. It's actually thin, but whatever. And so usually this T cell receptor complex and CD4 or CD8 are diffusing separately in the membrane, because proteins just diffuse in the plasma membrane. If MHC plus peptide comes around, you can see that the T cell receptor and CD4 or CD8 will both bind to that MHC plus peptide, which will force the cytoplasmic domains of these proteins into proximity with each other. We have induced proximity. This is supposed to show them far. They're not very far, but I can understand there's constraints of what you can draw in a textbook. And here they're close. And when they're close, LCK, which is a kinase, is able to add phosphates. See, here's a phosphate that your book shows. To the ITAMs of CD3. So now our CD3 ITAMs, these yellow rectangles, all get little pink dots because we have phosphates that have been added by our kinase LCK. All of these phosphates on the ITAMs allow for additional proteins to come in and bind. You can sort of think of this as being a change in the shape and the charge of the ITAM, now leading to another binding site. When I was in college, my senior year, my roommate who I had senior year is actually now a district attorney in southern New Jersey. And at one point she was like, you gotta explain to me what you're studying today. And so I was trying to explain some part of immunology to her. And I was like, actually, it's just all shapes. It's just about whether the shapes match or whether they don't match. And she was like, really? Why didn't you tell me that science was Sesame Street? Then I would have been a scientist. And so what you will see here, and throughout a lot of this signaling, is all we've done is we've made a new shape and charge in the cell by putting this phosphate on the ITAM. And so now we have a binding site for more proteins that we didn't have before. Our next protein is going to bind in this area. And our next protein is a protein called ZAP70. So ZAP70 is now going to be able to bind to those phosphorylated ITAMs. And so here you can see ZAP70 has this little section that can bind to the phosphorylated ITAMs. ZAP70 is also a kinase, and it's also going to be able to phosphorylate a number of different partners. So once we have the phosphorylated ITAMs on CD3, ZAP70 is able to be recruited, and it's able to start phosphorylating more targets. There are a bunch of other proteins that come in after ZAP70. I'm going to mention two of them, and I'm going to honestly tell you that they're interchangeable, although that, obviously that's probably not really true, but for our purposes, they can be interchangeable. These two proteins are known as LAT and SLP76. Yeah, Nadia.
This may be a stupid question. So
There are no stupid questions.
Is it anchored to CD4 inside?
It's closely associated with CD4.
Okay.
So it's CD4 coming close is what brings LCK close.
Okay. And then once LCK is activated, does it detach and go to the other side to phosphorylate, or does it just phosphorylate from one end?
It phosphorylates from one end. Some of this ZAP70 is going to be helping with too.
Okay.
I'm sort of trying to oversimplifying a little bit about which thing gets phosphorylated where with CD3. I just said all of it by LCK, but really ZAP70 is going to do some of it too.
Okay.
Great question. So once ZAP70 is doing some phosphorylation, we're going to get recruitment of these next two proteins, LAT and SLP76. And LAT and SLP76 are both part of the same class of proteins. LCK was a kinase. It added phosphates to things. It's an enzyme. ZAP70 was a kinase. It's an enzyme. It adds phosphates to things. LAT and SLP76 are what are known as adaptor proteins. Adaptor proteins are really all about the shapes. Because adaptor proteins themselves do not have any enzymatic activity. They are simply proteins that have a lot of docking sites for other proteins when phosphorylated. So you can see a nice version first at the bottom here. So if our adaptor, it's called a scaffold here, but it's the same thing. If our adaptor protein gets phosphorylated, get some pink dots added to it, it now is just a binding site for a whole bunch of other proteins. And it lets those other proteins come together, come into proximity in order to act on each other. It doesn't do anything itself. It just sits there and is a site for the others to come together. And both LAT and SLP76 are adaptor proteins. You can also think about the adapters that you use when you travel abroad. They basically are just changing the signal from a 3-prongy thing to a whatever prongy thing they have in that country. They're not actually doing anything to the electricity. They're just making the switch from one type to another in terms of the prongs. And so that's really what these adapters are for. LAT stands for the linker of activated T cells. And it's just linking these. And so we have LAT and we have SLP76, which just get phosphorylated and provide docking sites for lots of other proteins. And so this is where we have our adaptor proteins. The sort of interesting thing that can happen with LAT and SLP76 is that we get the proximity of lots of proteins, and we can start lots of different pathways. And so what I have to do now is walk you through a few different things that could happen. There are branches that happen after LAT and SLP76. So at the beginning, it's always LCK, it's always F70, it's always LAT and SLP76, and then we get some branching that happens from this point. So we're going to see a few different branches. The first branch is the one that students like the best. So the first proteins that can get turned on are involved with activating something called Ras. I'm not going to go into all the details of exactly how we're going to turn on Ras. You can imagine that LAT and SLP76 officially interact with Ras itself, because I'm not going to give you all the details of turning on Ras here. But before we get into Ras, I want to talk a little bit about other parts of this cascade. So let's imagine that we want to push a cell into cell cycle. We want our cell to divide. What's that process called? What is it, Jordan? Mitosis. And a stimulus that induces mitosis is sometimes called a mitogen. And if you have proteins that are turned on in your cells, when you stimulate them with a mitogen, you might call them mitogen-associated proteins. Or MAP. There is actually not a protein in your cell called MAP, but this is going to make our life a little easier to think about, pretend there is a protein in our cell called MAP. So all of these proteins, these mitogen-associated proteins, get turned on by phosphorylation. If you were in charge of the world, what would you call a protein that phosphorylates MAP? Yes, Malik. You might call it a MAP kinase. Or MAPK. And in fact, when you think about MAP kinases, it turns out that MAP kinases are not naturally active. MAP kinases require a signal in order to get turned on. In fact, they require phosphorylation. If you were in charge of the world, what would you call something that phosphorylates a MAP kinase? Yes, Emilio.
MAP kinase.
You would call it a MAP kinase. Okay, MAP kinases are not automatically active. They have to be turned on. They are turned on by phosphorylation. If you were going to be in charge of the world and you were going to name a protein that turns on a MAP kinase by phosphorylation, what would you call it? What would you call it, Leah?
A MAP kinase.
You would call it a MAP kinase. Or MAPKK. MAPKKs are not always active in the cell. They have to be turned on. MAP kinases are turned on by Ras. And so if you remember that this pathway is called the Ras-MAPK pathway, you're done. In terms of memorizing, that's why students love this one. In your cell, there are multiple different versions of MAP kinases, MAPKKs, and MAPKs. So I am going to tell you the names of a specific MAPKK, specific MAPKK, specific MAPKs, going through this process. But they're all part of this general group. So in T cells, Ras turns on a MAPKK that's known as MEK. Wait, no, that's RAF, sorry. Whew, that was bad. All right, so Ras turns on RAF, which is our MAPKK. Our MAPK in this case is called MEK. And our MAPK is going to be called ERK. ERK phosphorylates a protein that you can think of as a mitogen-associated protein. In fact, it's a transcription factor that is known as Phos. And so this is a transcription factor. There is a separate MAP kinase pathway that leads to the turning on a different MAP kinase that's called JUNK. And JUNK turns on a different transcription factor called Jun. JUNK is Jun kinase. So it turns on phosphorylates Jun. And when Phos and Jun are made together, they turn into a transcription factor called AP1. So one of the things that happens downstream of our adapters is we turn on Ras and the MAP kinase pathway to make AP1, which is one of our goal transcription factors before. And you can see that same thing from the old version of your textbook. We have Ras, then RAF, then MEK, then ERK. We get Phos and Jun together to be AP1, and transcription is on. So this is one of the things that happens downstream of our T cell receptor. But as I said, other things can happen downstream of the T cell receptor as well. I'm going to tell you about something that I'm calling here kind of pathway 2. It's really going to have a 2A and a 2B. In real life, there's also like 3 and 4 and 5 and a whole bunch of other ones, but we're going to do 1, 2A, and 2B. The other protein that I want to tell you about that's activated downstream of LAT and SLP76 is a protein called PLC gamma. To really understand what PLC gamma is, I should tell you what PLC stands for. That's going to be the first thing that's going to help. PLC gamma stands for phospholipase C gamma. If you see the word phospholipase, what might you guess is the activity of this protein?
phospholipids?
Cleaves phospholipids. ACE, it's an enzyme, and cleaves phospholipids because it's got phospholipid at the beginning. So phospholipase C gamma cleaves phospholipids. It specifically cleaves the phospholipid that's shown here, called PIP2 or PI45 bisphosphate. I always call it PIP2. What you can notice is that PIP2 has two fatty acid tails, usually that are embedded in the cell membrane. And then it's got this nice head of the phosphate charge group there. And phospholipase C cuts PIP2 into two molecules, one called diacylglycerol, which keeps the fatty acid tails and stays by the membrane. It's usually known as DAG. And another, IP3, inositol 1,4,5 triphosphate, which is this head group. And as I said, there is going to be a 2A and a 2B, and that's because we can follow what happens to DAG, and we can follow what happens to IP3. Immunologists care a lot about IP3. And so that one is really, really, really famous. I always feel bad for DAG, that no one ever talks about it, and so I'm going to talk about DAG too, because I just feel bad for it. But IP3 is the super big one. So we've got to follow each of these two products, DAG and IP3. And we're going to follow IP3 first. IP3 is going to move through the cell. It's going to bind to a receptor. If you were in charge of the world, what would you call this receptor? Yes, Molly?
An IP3 receptor?
Molly would call it the IP3 receptor. And in fact, the name is the IP3 receptor. The IP3 receptor lives on the endoplasmic reticulum. And so when IP3 binds to its receptor on the ER, it results in calcium ions being released from the ER. Your cell works really hard to keep the calcium concentration in the cytoplasm very, very, very close to zero. There's almost no free calcium in the cytoplasm. All of the calcium is either put into the ER or outside the cell. And so you can have a great signal if you just release the calcium back into the cell. That can be a very strong signal where that calcium, as soon as basically you open the channel, it just floods from high to low concentration. You get a really nice signal. So IP3 opens the gate, lets the calcium out. And you can see that in this figure as well. Calcium is the little blue dots. At the beginning, there are no blue dots in the cytoplasm. They're all in the ER and outside the cell. But when IP3 binds to a receptor, we open the channel and the calcium goes into the cytoplasm and out of the ER. So once we have additional calcium in the cytoplasm, it can bind to a calcium binding protein. Our calcium binding protein is called Calmodulin. Calmodulin has a number of different effects. You've likely heard of Calmodulin in some neuro classes, for example, because it's really important in neuroscience as well. But in either case, it's binding to calcium. And so Calmodulin is going to be able to bind to calcium ions, and that's going to induce a conformational change to allow it to bind a target protein and turn that target protein on. Apparently, it's going to be a little worm with sunglasses. What's going on here? So this is what's going to happen with calcium and Calmodulin. Once calcium binds Calmodulin, which here is a wrench. So calcium binds to Calmodulin, it undergoes a conformational change and turns into a wrench. Calmodulin is going to be able to bind another protein called calcineurin. Calcineurin is going to interact with a protein called NFAT. NFAT normally in the cell has phosphates on it. Phosphates are still pink dots. So we've got pink dots all over NFAT. When calcineurin is active, calcineurin is able to remove those phosphates from NFAT. So we now can see NFAT has no phosphates where it had phosphates before. has its phosphates removed, it is allowed into the nucleus to do transcription. So NFAT normally sits in the cytoplasm covered in phosphates. When it's covered in phosphates, it's not allowed in the nucleus. Calcineurin removes those phosphates from NFAT, thus allowing NFAT into the nucleus to induce transcription. Happy, NFAT's one of our other transcription factors we need. So now we got 2 out of our 3. Yep, Nadia.
So, in this case, phosphorylated NFAT is deactivated?
Is off, yeah. So phosphorylated NFAT is off. It's non-phosphorylated NFAT that's on. Immunologists talk about this pathway all the time. This is a super, super, super famous pathway in immunology. And the reason this pathway is so famous is one of the first big drugs that affected the immune system, Cyclosporin A, acts on this pathway. So Cyclosporin A is shown here as this orange little thing. Cyclosporin A interacts with the cell protein and keeps calcineurin and calmodulin from interacting with each other. So even when our T cell is getting signal, calcium gets released into the cytoplasm, calmodulin's got it, but we can never turn on our T cell because we block that last step of calcineurin activation and we can't ever take the phosphates off of NFAT, and NFAT is stuck in the cytoplasm, sadly, forever. This drug is the reason why transplantation is a thing today. This basically turns off T cell activation, and this is the drug that allowed transplantation to basically become possible because we could immunosuppress patients with cyclosporin. Cyclosporin is used to immunosuppress patients in a number of different ways. It's a pretty blunt kind of thing. It's turning off signaling in all T cells because it's stopping them all from doing the signaling pathway, but we couldn't do transplantation before cyclosporin. So cyclosporin is a huge deal, which is why of all the T cell signaling pathways, immunologists, they know the IP3 part of this pathway. DAG also does stuff. I don't want you to feel like DAG is off on its own, not doing anything. So DAG is able to turn on some additional signaling, and eventually DAG leads to NF-kappa B activation and movement into the nucleus. And so all together, we start with our T cell receptor binding LCK. We get phosphorylation of our ITAMs. That recruits ZAP70, which does some more phosphorylation. We can then get LAT and SLP76 recruited and phosphorylated. This will bring in the Ras pathway. We'll get Ras-MAPK activation, and Ras-MAPK activation will turn on AP1. We'll get PLC gamma activation. PLC gamma will cleave PIP2 into IP3 and DAG. IP3 will induce calcium release from the ER, which will then turn on calmodulin and calcineurin, leading to NFAT activation, and NFAT will go to the nucleus. We will also get DAG to turn on NF-kappa B, and NF-kappa B will go to the nucleus. And in the end, happily for us, we now have NFAT, AP1, and NF-kappa B all working together, and we can turn on IL-2, which is the first part of really starting our T cells response. Yep, Molly.
Is NF-kappa B activated by phosphorylation?
It is not. So the way that NF-kappa B is activated, this is oversimplified. NF-kappa B actually has 2 parts, and there are different combos you can have and all that business. But we'll say that's NF-kappa B right now. NF-kappa B has a site that lets it go into the nucleus, nuclear localization signal. But it's normally covered up by an inhibitor called I-kappa B, the inhibitor of kappa B. So normally this covers the nuclear localization signal. What actually happens is that I-kappa B gets phosphorylated, And that phosphorylation triggers I kappa B to come off NF kappa B and go to the proteasome. So I kappa B gets degraded so that it can no longer inhibit NF kappa B. And NF kappa B is now allowed into the nucleus where it was not before. There's a combination of 3 kinases that do this phosphorylation, IKK alpha, IKK beta, and one named Nemo, which took them a long time to find. And yes, that's how it got that name. Yeah, Malik.
Is that what DAG signaling does, or?
DAG signaling acts on a kinase that turns this stuff on. The DAG, going through the rest of DAG, it's a little more complicated, but I always feel bad leaving DAG out. One thing that you might want to think about here, and you will see this if you look at some of the old exams, is that there are kind of some questions about receptor proximal versus further downstream signaling components. So, for example, let's look at this for a second, and you will find on old exams questions like this. So we've got our 3 transcription factors. Should probably put up the one that has all. We've got our 3 transcription factors. So imagine that I have a drug that blocks LCK. Okay? Which of the 3 transcription factors are blocked if I block LCK? What do you think, Manny? All of them. Why all of them? Yeah, LCK is one of those receptor proximal molecules. And so if you get rid of LCK, the whole thing downstream is done. Now let's think about cyclosporin. If we use cyclosporin, which transcription factors are blocked? Yep, Mario.
Only NFAT.
Only NFAT. Why only NFAT?
Because it only blocks the interaction between calmodulin and calcineurin.
So, cyclosporin is acting here between calmodulin and calcineurin. So NFAT's not going to get turned on, but AP1 and NF kappa B are still going to go just fine. And so it can be very important for you to distinguish between whether you're thinking about a receptor proximal protein or a receptor distal or a further downstream protein when thinking about signaling, because blocking them can have different effects. Blocking something that's a little more downstream might be a little more specific, as opposed to blocking something upstream, which is going to stop everything altogether. And so that's a key thing that you will see on a number of those different processes. NFAT, AP1, and NF kappa B together transcribe IL-2. As I said before, these are not the only things that actually happen downstream of LAT and SLP76. There are also big changes in terms of other types of transcripts and other aspects of chromatin in these cells. There are also big changes in the cytoskeleton of the cell, as well as pushing the cell into cell cycle. So we're going to get survival, we're going to get proliferation, we're going to get effector function, we're going to get a whole bunch of things going on, as well as some cell movement, some rearrangement of the cytoskeleton. So there's actually a lot going on downstream of LAT and SLP76. These are just the main ones I want to tell you about. So once we've had our 3 transcription factors, we can finally make IL-2. And IL-2 is made by T cells that have been turned on. So you can see our early T cell is not making IL-2. There's no little arrow leading to one of these blue dots. However, our T cell that has seen antigen now makes IL-2 and actually feeds itself with the IL-2 receptor, which is more going to be a Wednesday thing. IL-2, when it was originally cloned, you can imagine it was the second cytokine. When it was originally cloned, its name was not IL-2 because we hadn't come up with this whole cytokine nomenclature thing yet. IL-2 was originally called T cell growth factor, TCGF, because it's the thing that makes T cells survive. If you want to actually grow T cells in culture, for example, you have to give them IL-2. T cells cannot survive and cannot divide and cannot go through cycle to do this expansion without IL-2. So IL-2 production is really going to be the key to feed these T cells to let them do the rest of this process. Our cell is also going to change as a result of this process. Originally, our naive CD4 cell is not going to be all that great at producing cytokines. However, once that cell becomes activated, that cell is going to become a helper T cell that's really great at cytokine production. So it's going to become more specialized. Therefore, we don't call a naive CD4 cell a helper T cell because it doesn't have the ability to help yet. It's a naive CD4 cell. Once that cell is activated, it becomes a helper T cell. Similarly, a naive CD8 cell is just a naive CD8 cell. Once that cell has become activated to go through this whole process, that cell is now going to become a CTL, an activated cytotoxic cell. This makes particular sense when we think about CD8s because you don't really want this original CD8 cell to kill this dendritic cell. Then you would not present any more antigens and turn on any more CD8 cells. You only want that CD8 cell to kill when it gets back to the periphery where the infection is happening. One can make some other arguments about what's going on with whether the cytokines would be useful in the lymphoid organ or not for the CD4. But for the CD8, it's real obvious that you wouldn't want to kill anybody in the lymph node. And so, in reality, though, there are additional additive signaling processes that are happening that add on to this normal signal. These are coming from signal 2, which is our co-stimulator, and signal 3, which is our cytokine. You guys are really going to like the co-stimulator. The reason why you're really going to like it is when we talked about T cell development, you freaked out a little bit about, you thought we were all going to die of autoimmunity. And the co-stimulator is the answer to a lot of the questions you had that day. The other thing about the co-stimulator is that the co-stimulator is involved in the Nobel Prize this year. So the co-stimulator is pretty cool. And we also finally need cytokines to fully get that T cell turned on. I'm not going to go through the signaling with those two in quite as much detail. There'll be a little bit of cytokine signaling, but not a whole lot of the co-stimulator signal. But we are going to talk about how those interactions happen and what that does to the T cell next time. And then we'll start with the, oops, wrong way. We're going to start with how functions work for CD8 T cells, because that one I can go over pretty quickly. It takes a while to go over the functions of CD4 cells.